Just a couple of, sorry, yes. Just a couple of reminders and housekeeping before we introduce our lovely speaker, Professor Laura Vella today. We have our YouTube channel now, so if you want to find previous recording or this recording, please subscribe to our YouTube channel. The link is on the screen. And if you haven't already registered for the conference, you can do so for the upcoming conference on the 24th to 27th November at our ANZSEV website. And also this session, although it's for 1 hour, it wouldn't run for full 1 hour. It would be around 35 to 40 minutes with 20 minutes speaking time and 10 minutes discussion time. So it will finish a little bit early. And if you have any questions, please put them in Q&A chat box. Now I would like to introduce Poppy to welcome our speaker for the day. Poppy, you can.
Thanks, Tricia. Associate Professor Laura Vella is the head of the Extracellular Vesicle team at the Florey Institute and the University of Melbourne. Her work focuses on defining the molecular content and function of EVs and translating these insights into EV-based diagnostics. She's the leading expert in the field with more than 20 years' experience advancing understanding of the role and application of EVs in human health and disease. She currently serves as the chair of the ANZSEV Tissue Task Force, chair of Research Integrity Advisor Network at the University of Melbourne, and is advisor to biotechnology companies in the sector. Thanks for joining us, Laura. We look forward to your talk.
Thanks so much, Poppy. I will just share my screen. How is that? Can you see my screen?
Yeah, that looks great.
Fantastic. Okay, so thank you so much for inviting me. So in the next 20, possibly 25 minutes, I have with you today, I'm going to talk about isolating extracellular vesicles or EVs directly from solid tissue. So the reporting recommendations from the International Society Solid Tissue Task Force was published last year in 2025. And so I'm going to talk about those recommendations and touch sort of briefly at the end on how my team applies that method to study the brain. So I have to apologize in advance that the talk is really text heavy in order to really convey the recommendations. Okay, so this obligatory EV slide. So everyone here is likely an EV enthusiast, so I'll keep the intro to one slide. I've probably offended a lot of people by keeping the slide so simplistic and not really including EVs from different organisms or cell types or even within the same cell. So we know they're highly heterogeneous and there's many different vesicle populations. I've really used this as a simplified figure to highlight, I suppose, some of the trafficking pathways that my team and I are interested in. So, we know, for example, if you've got a protein at the plasma membrane, you can get endocytosis and incorporation of that protein at the early endosome. You can get incorporation, you get conversion of the early endosome into a late endosome, and you get inward budding of the bilayer of the multivesicular body. You get formation of these intraluminal vesicles, which upon fusion with the plasma membrane, so they can either be degraded or upon fusion of the plasma membrane, these small vesicles are released into the extracellular space. They're often referred to as exosomes, really depending on how confident you are with assigning the origin. So the figure also shows budding of the vesicles from the plasma membrane, in which these are sort of larger vesicles. So why do we care about EVs and why has the field really, since I started, exploded as much as it has? Three main reasons. They're functional mediators of cell-to-cell communication, and they can transfer behavior between cells. Diagnostically, they can act as readouts of tissue and disease state and therapeutic response. And they offer a way to detect disease-associated signatures in biofluids. And then therapeutically, they're being explored as delivery vehicles. Okay, so talk outline. So why EVs from solid tissue is what I'll touch on. Secondly are recommendations and guidelines for isolation of in situ EVs, and I'll comment as to why we're looking at EVs in situ. And then application of that methodology to the brain in health, aging and disease, and touching on what my lab has done and is doing. Okay, so most when people are looking at EVs, generally they're looking at EVs isolated from biofluids. And as we know, EVs have been isolated from every possible biofluid now. And, you know, there's the common blood, urine, spinal fluid. So they're accessible. Whereas EVs coming from a solid tissue are separated directly from, for example, adipose tissue, heart, liver, kidney, and in my lab in particular, we look at the brain. And so the vesicles are separated from fresh or frozen, you know, a piece of tissue. So there's no culture or expansion step. So these EVs are released basically from that tissue, and they're coming from all cell types that are within that tissue. And the reason I suppose I stress that we're looking at these in situ EVs, and that's what I'm talking about today, and that's what the recommendation paper is on, is that culturing the actual tissue, so explant, is, I suppose, a different strategy. And, you know, while some of the guidelines I talk about are applicable, the talk will be on in situ EVs. Okay, so a bit of a brief history. So Perez Gonzalez, who was in Efrat Levy's group in the US, first established a method in 2012 where they enzymatically basically released EVs from fresh and also frozen human and mouse brain. And then in 2017, we came along and sort of refined it further using collagenase to reduce the cleavage of surface proteins, using modified gradients and really exploring the protein and RNA content. And then from 2019, the approach was used on tumour tissue. So Jan Lotville's group, particular Rosella Cristelli, who's on co-chairing the task force, we're looking at melanoma and a variety of other different tissues. So 2019 onwards is where we saw the field kind of explode a little bit, and whereby I've just mentioned a few different species here. You know, the common mice and rat, but then there's been some really unusual species as well. We've got freshwater mussels and ticks, which is kind of cool. And, you know, really different tissue. So bone, salivary glands, cochlea. And, you know, I think we're now seeing those isolations and that examination of composition and function kind of starting to deliver in regards to understanding the biology. So, and findings that we know some of the proteins now that are regulated, that regulate EV release, biomarkers that can track disease, and you know, really clear evidence that these tissue EVs can drive tumor growth. Okay, but pulling EVs out of tissue introduces a lot of confounders that you don't get with biofluids, and there's three big ones. So I've got them listed here. So the pre-analytical variable, so this is before you even start, you know, looking at EVs. So you've got different species, storage, and I'll get onto a little bit more of this, but post-mortem interval, disease state, all change yield your ability to isolate the EVs in the first place. Dissociation artifacts, so you're dealing with, you know, intact cells, some of them are going to be lysed, but you can, and you've got potentially that mechanical and enzymatic digestion also resulting in co-isolation of organelles and generation of EV mimetics. So expertise as well, really, it does depend on who is doing the isolation, and this is really particularly at the start when individuals are learning, and I suppose, and you know, these really confounders, I suppose you would call them, are the reason why the task force set out to put this paper forward. Okay, so the task force organised its recommendations into 4 stages, which is basically the workflow from starting from a tissue to having your EVs and characterising them. So I'll take you through each of the 4 stages. The last 2 aren't going to be particularly new to anybody, but really the challenges are with these first 2 steps. So I'll talk about sort of best practice, what to probably try and avoid, and then some of the questions that we still can't answer. Okay, so stage 1, solid tissue collection, storage, and handling. So this is before you even touch an enzyme, start to dissociate. And for human biospecimens, typically they're coming from surgical specimens, they're surgical specimens coming either straight from surgery. In this instance, we sort of recommend removing as much blood as you can by simply sort of rinsing in PBS or saline. Freezing liquid nitrogen straight away, or if you're going to isolate those EVs immediately, then keep that tissue on ice until you're ready to go. Where it starts to become a little bit challenging is with biobank specimens, in that each biobank can function quite differently. And so often some of them don't follow standardized collection procedures. So I recommend, if you're using a biobank, is to use one that follows that standardized BRISC or NCI collection procedures. And there's a couple of obvious things like avoiding repeated freeze thaws. If you're working with animal tissues, life is a little bit easier in that you've got a little bit more control of that tissue. Some people choose to perfuse the animal, others choose not to. I don't think there's any right or wrong way here. The only suggestion we'd have is that if you do choose to perfuse, try and make sure it's automated. So basically you've got control of the pressure and the flow. So outstanding questions, and these are pretty big, is that how much do storage buffers affect recovery? So if the tissue is going into a storage buffer, what storage buffer should it be? Often in our case, the tissue is coming as a piece not in storage buffer. How does that affect recovery? Can we work from fixed tissue? And there's some evidence coming out to suggest that we can. And then how do we minimize blood contamination? Okay, so stage 2, and this is again one of the really challenging parts. And I suppose the part that, when I was in the lab and the team now, we get quite stressed when we're doing it, because we try and do it as quickly as we can, and really trying to preserve the integrity of the material as much as possible. Okay, so when I say dissociation and releasing those EVs, it is basically trying to dissociate the tissue as gently as possible, so the EVs are then released into what will be your buffer or your supernatant. And the goal is really to keep those cells intact. There will, of course, be some that are lyes, but you want to keep the cells as intact as you can, but also ensuring the surface profile of those EVs and any corona are preserved as much as possible. And so we recommend using a really mild, very brief enzymatic digestion. So slice your tissue quite thinly and then use a targeted enzyme. And then basically inhibit that specific protease the moment you're done. So most of the time you're looking at generally 25, 37 degree incubation for it might be 10 to 25 minutes, and it really depends on the tissue you're working with. And that protease, and then a protease inhibitor needs to go in and to basically inhibit that digestion. Otherwise you will just get continued digestion. So keep everything cold, handle for a minimum amount of time, and then run one identical protocol across all your samples. It'd be lovely to do all your samples in parallel, but this is generally impossible. We would suggest avoiding homogenization and vortexing. So there has been there is a way in which you can basically some people have taken the tissue and they will just homogenize and vortex it. in that respect, you're not necessarily looking at the EVs in the microenvironment because you are, destroying that tissue per se. Okay, so what we don't know is, and again, really important questions we need to answer is how do we actually quantify the degree of digestion? So how do we know we've released all the EVs? How do you confirm it? And what's the sweet spot between making sure you've got enough dissociation and then preserving that integrity? Okay, separation and enrichment. So really the only thing that's kind of different here is the start of the separation and enrichment process in regards to, the later steps are sort of common to isolation of EVs. So really it's as soon as possible, as soon as that digestion is complete, remove your supernatant, your EV containing supernatant for what is going to be your cells and all your debris, which is a lot. So get rid of it as quickly as you can. And a lot of people will use basically an optional filtration step or pretty much a cell strainer to get rid of a lot of the cells and bulky material. Others use a low speed centrifugation. Either way, both of those methods are fine. And then separation after that is generally with a combination of methods. And as we know, they each have their unique, trade-off. And I won't go into what they are because we all sort of are familiar with the different isolation enrichment methodologies. So one open question that some people raise is whether the separation needs to be tissue specific. I don't think it does. There's no reason why it needs to be tissue specific. What does need to be specific is actually the dissociation step. So if you are starting out in the field and you are looking at a particular tissue, particular species, you need to optimize the digestion conditions for your particular tissue because it's going to be different depending on the batch of enzyme you have, how the tissue's been stored. And so you really need to make sure you characterize what you've isolated and ensure that all your conditions are correct before you move on to, thoroughly investigating those EVs. Okay, stage 4, characterization and assessment. All right, I'm not going to touch on the first 2 points here because they're common to all the EV characterization steps. But specific, I think, to the in situ EVs is sort of probing, and you can do it Western blot or even proteomics, is examining the cell of origin markers. So basically tie the EVs back to the source they've come from. The other suggestion I have is just making sure that your surface proteins actually survived the digestion. So, for example, the prion protein, it's cell surface, GPI anchored, too much digestion, and it will actually be shaved off. And if we want to really look at the entire EV, we want to make sure we don't digest its surface profile. Another thing I will mention is, I suppose, just a word of caution when it comes to particle counts. So they can be kind of confounded just because of the variability that exists in different tissues. So if you take Alzheimer's disease versus control, that AD brain is going to be quite atrophied. So it's going to digest very differently to a neurological, control brain. So comparing the raw particle counts can kind of lead you a bit down a garden path because what you're measuring is actually could be how well the tissue's actually digested. So that efficacy, not necessarily the biology, if that makes sense. If anyone's got any questions, they can ask me at the end. Okay, so I want to turn now to our own research program on solid tissue EV, specifically brain-derived EVs. What we'll do here is I'm going to basically take you through some, I suppose, a bit of an overall or a bit of a snapshot, and then close with a couple of slides on sort of some really recent data that that we have been generating. Okay, so a word, I suppose, on the clinical problem and what really motivates us at the Florey. And So we're working on neurodegenerative diseases, among others, and these diseases are characterized by a progressive loss of neurons and their connections. We've got Alzheimer's and Parkinson's here as two examples. Both are rising sharply with age. Alzheimer's disease, as we know, get cognitive decline. It's the most common cause of dementia, with about 70% of cases of dementia, which is that umbrella term, actually being Alzheimer's disease. Parkinson's disease is recognized as a movement disorder, but it's also a multi-system disorder. So despite those diseases actually being quite, different, I suppose, their clinical differences, they share, I suppose, major hurdles or challenges, I think is probably the right word. And, the therapeutic landscape is evolving, and, treatments that actually can robustly treat either of these diseases. So there's nothing that exists for Alzheimer's disease, for Parkinson's disease that is actually disease modifying. And then there's been a couple of therapies that have come out in the last year for Alzheimer's disease, but they've got their challenges as well. So the molecular pathways that actually drive disease onset and progression remain sort of, not completely understood, incompletely understood. What the field really needs is a better understanding of which biological pathways are disrupted, what actual changes are happening, and those really early changes and how we can measure them. And we need accessible biomarkers. So for Alzheimer's disease, in the next probably year, you'll start to see diagnostic tests come through for Alzheimer's disease, the very first. But any biochemical, there is no biochemical blood-based biomarker for Parkinson's disease currently. Okay, so this is where for us brain tissue-derived EVs become really valuable. So they provide us a way to basically interrogate these disease-relevant cellular pathways really directly in the brain microenvironment, and we can start to identify the molecular signatures that ultimately might be, translated or are translated into those more accessible biofluids. Okay, so this is actually a really old slide, but we still, the process is all really similar. So in our lab, we work with human and rodent brains, and it's this really first step that is key. So depending on the disease that we're interested in, we'll look at the frontal cortex, the motor cortex, different regions depending on the disease. We'll take a piece of the tissue, we slice it and digest it with collagenase. And then, really we basically follow the standard EV isolation preps, standard characterization in regards to looking at density, size, morphology seen here by electron microscopy, whether those EVs contain RNA's protected RNA, and then characterize them via immunoblot, looking for enrichment of EV markers and depletion of non-EV markers. So over the years, we've used a multi-omics approach, but the focus more recently or always has been, I suppose, proteomics. Okay, so these are some of the research areas my team has or is working on in both human and rodent tissue. And I suppose what I wanted to touch on was that there's been a common finding both, I suppose, with cell models in vitro and also these in situ EVs, and that I find that the field you're always reading that the EVs mirror the content of the parent cell. And I really don't think, based on what we've found, they aren't a general reflection of the whole cell, and they're really a readout of particular intracellular pathways. Okay, so coming back to my sort of very short, my biogenesis slide. So when I say EV content's not simply a reflection of the whole cell. So we know, small EV formation is linked to intracellular trafficking. So you've got the quality control pathways, endosomal trafficking, autophagy and lysosomal clearance, and mitochondrial homeostasis or dyshomeostasis that feeds into these pathways. So these we know that the autophagy pathway interacts with the MVBs, mitochondrial derived vesicles, and content can enter the endolysosomal and EV pathway. So basically any disruption in those pathways, that altered cargo gets incorporated into those small EVs. And that's why in our lab we've been particularly interested in them, because that EV cargo is not a random sample of the cell. It's selectively packaged and a concentrated output of those pathways. And for us, that's important and key, and why we in particular try and look at these particular EV populations. So in Alzheimer's disease, you've got, it's been known for decades that some of the earliest cellular dysregulation, I suppose, is in the autophagy lysosomal pathway, and in Parkinson's disease, it's mitochondrial dysfunction and impaired endolysosomal trafficking. So our work has really shown that these disruptions are reflected in the composition in proteins. We see this in proteins, lipids, metals, and I suppose this is an important point, is even when those same changes are not readily detectable in the whole cell or even the tissue. And I think what that means is it reflects, I suppose, the specific enrichment of this specific cargo. And consequently, those EVs are providing a higher sort of signal to noise readout. Okay, so how are we going for time? Have we got another 5 minutes, Trisha?
Yes, you can go ahead.
Yeah. Okay, so I now want to touch on some work from Ella Parks in the lab, and this is sort of hot off the press and really quite new. And it's related to Ella's aging study. And so one of the advantages of working with the solid tissue EVs is that we can capture the vesicles released in their native environment. And that's great, but it's also challenging because you're looking at the whole pool and you're missing out on actually examining, cell-specific EVs within that pool. So Ella's started to begin to resolve this in relation to the brain. And what she's done is developed a nanoflow approach to analyze the brain tissue-derived EVs at basically a single EV level. And so there's two basically steps. She labels the EVs with CFSE, which is basically a pan dye, I suppose, and it fluoresces once it goes inside those EVs. And then, it helps you, I suppose, figure out the background from the noise. And then she applies antibodies against brain-specific cell-associated markers. So a caveat to this slide is that, we know small EVs are really painful to detect via flow and nanoflow, but nanoflow is certainly of benefit over flow. So the approach really requires careful controls, instrument settings, and this has really been hammered home. And I wasn't able to attend the last ANZSEV talk, which focused on flow, but it would have really come across that we need to be really careful in the way in which we control these experiments. So I don't have the time to go through all of the controls that Ella's done, but if you're interested in using nanoflow for EV analysis or want to reach out to find out how these experiments were done, please reach out to me and I can put you in contact with Ella. Okay, so what Ella's done here is basically taken the mouse brain tissue, applied this approach, and then used an antibody panel. And here we're interested in the EVs coming from neurons, astrocytes, oligodendrocytes, or microglia. And what you can see here is that basically astrocytes are making up, relative to the proportions of the other EV populations that we're looking at, that the astrocytic EVs are making up the largest portion here at about 43%. So having established the method, Ella's now gone on to applying it to looking at the aging brain and how these marker populations basically shift with age. Okay, so because the readout is quantitative, we can ask how and measure basically how the populations change. And so the question is, who cares? Why do we want to know how the populations change and their content? And basically, we know aging's associated with cellular senescence, and there's a variety of different impairments that happen within the cell that are then, and the cells basically, they acquire what's called a senescence-associated secretory phenotype. So they release a lot of inflammatory signals and factors, and that then they are known to influence their neighboring cells. So EVs are part of the secretome, we know they mediate intercellular signaling, but how actually aging alters the brain EVs and their cargo and functional effects is really not known. So Ella's taken a cohort of wild type animals, and they basically is a cohort spanning from 3 to 18 months. She's isolated the bulk EV, so similar approach to what was shown on the previous slide. Looked at 4 animals at each time point and then looked at those same specific EVs by flow. So she's just finished this analysis here, and what she's looked at is EVs coming from neurons, astrocytic cell, astrocytes, microglia, and oligodendrocytes. And, we start to see that basically you get an increase in some of these EV populations with age, which does kind of fit in regards to, we know that some of these cells basically start to adopt a reactive state with aging. And we know that cellular dysfunction increases vesicle release. But what I like about this approach, did it come up? Yeah, it has, cool. Is combining the EV phenotyping that Ella's doing with proteomics. So really defining the cargo of each of those EV cell populations and asking whether those cell-associated EV signatures change with age. So in partnership with the Melbourne Cytometry platform, Ella's now trying to actually fact sort out these EV populations. And the idea is, if we can detect them, hopefully we can sort them, then look at their individual cargo and function. So the approach is technically challenging and probably a bit of a pain in the butt, ask Ella, but it brings us, I think, close to, as possible, to a physiological readout of the changes that are happening in this, in the brain and the central nervous system with aging. And I think that level of insight is only possible because you can isolate those EVs direct from tissue. Okay, so just in summary, in regards to the recommendations, there's a lot of pre-analytical variables, tissue species differences that can affect your solid tissue EV quality and composition. Annoyingly, but it has to be done, is, every time you're starting with a new type of tissue or species, organ, is you need to optimize the enzyme type, concentration, and duration. And unlike EVs from bodily fluids, we think that those solid tissue EVs offer a direct representation of the microenvironment and allow for investigation of those tissue-specific, I said it the wrong way around, these tissue-specific pathology, I suppose, by analysing the EV populations originating from those tissues. So if you haven't already, have a look at the recommendation paper, and if you've got any questions, shoot me an email. Okay, and then just in closing, I would like to thank all the members of the Tissue Task Force, particularly my co-chairs, Rosella and Lucia, and all members of the EV Lab, past and present. And I've spoken, a lot probably at the end there about Ella's work, but there's been a lot of key members, particularly Kate, that have contributed, and our funding partners. And thank you. I'm happy to take questions.
Thank you very much. That was fantastic. We learned a lot. And feel free to put questions in the chat box for anyone that has, or you can also unmute yourself. I think I will go first while people are still putting questions. I don't have any solid tissue EV background, so it could be a really, really simple question. But how do you tell apart these like intraluminal vesicles from EVs when you're digesting all the tissues, or these vesicles inside the cells versus like extracellular in terms of their signature?
You can't. So that's the tricky part, is an intraluminal vesicle is basically a small EV before it's outside the cell. So that's why it's so important to be really as confident as you can be in how the tissue has been stored, collected, and then those digestion steps. So it's not guaranteed. This is probably the dirtiest prep that you will do. The goal here is to make it, to basically apply as much rigor as you can and use the best, the highest quality tissue source you can to limit the amount of intraluminal vesicles that you would be examining relative to secreted vesicles.
Does it depend on like you measure the cell lysis and then you can kind of determine what you're getting from inside the cells versus outside?
Yeah, so I mean, you can. So you could do, you can do flow, for example, or have a look at trypan blue staining, for example, to have a look at how intact the cells are and then make that relative to the EVs you're looking at. It's not routinely done, but it is one of the suggestions moving forward that we should have a standardized way of being able to tell how much dissociation we've got and then how much cell lysis we've got, because there is going to be cell lysis. It's just a matter of what's the right way of measuring how much we've got.
Thank you. And just one more question. Which flow machine do you use for your
Cytoflex Nano. Ella, I hope I've got that right. I'm pretty sure that's it.
Okay, thank you. Anyone has any questions?
I have a question, if it's okay. Hi, Laura. Really nice talk. Thank you. So I guess kind of following on from Darisha's question, where it's really important to optimize our isolation, disassociation. What kind of things do you look for when you're, so say you have 2 or 3 methods and you're comparing them? What are some of the things that you look for and you say that's better than the previous method? Has it to do with antibody staining or signal?
Yeah, so commonly, so that's what we initially, when we started out in this, we were comparing, for example, the proteomic profile, or how sensitive they were to RNase digestion. The easiest and the quickest way is basically for us is Western blotting. And so we basically Western blot, we compare the profile of a lot of those proteins. We compare, say, the total brain homogenate with what is, and then compare that with the palette of our low speed spin with our EVs, and basically just make sure we're not losing that signal over time. And so yeah, look, the Western blot has been the most effective. And so what we've done, for example, collagenase, painfully, every single batch of it is different. So what Kate did in my lab, and thank her for doing this, is we got in, I don't know, it might have been 12 batches of collagenase. And so she treated tissue pieces with different amounts of collagenase, and then looked at the profile of the resulting vesicles after that digestion, and also the resulting brain, the brain homogenate with and without that enzyme.
Thank you.
I think Leslie has a question.
Hi, Laura.
Hello, Leslie.
So it was really nice to see your mice work in the aging model. And obviously we're always interested in neurodegeneration, but I'm curious, what are your thoughts in looking in all of the downstream work that you're doing in that model with neuro and brain development, like in terms of, childhood disorders, etc., that happen at early onset, and then having it go through to the aging brain?
Yeah, so look, we haven't yet, so we'd love to do it. We're lucky at the Florey, we do have a lot of neurodevelopmental models and also models whereby we're looking at, environmental exposure and the effects or impacts of environmental exposure on, progeny in regards to the animals. So we haven't done that yet. I would like to. It's just a matter of resources. So yeah, if you can tell NHMRC to give us a little bit more money, then that's certainly an area we'd like to look at.
Thanks.
Hi, I have a question as well. I was just wondering, you mentioned kind of how different the structure can be of the brain with Alzheimer's or Parkinson's, and then also I imagine throughout different stages of maturity. How do you account for those differences when you're developing your protocol?
Yeah, so that's a good point. So I suppose the first thing is report everything. So we basically record the amount of EV particles, the amount of protein we get per weight of tissue, the amount of protein we get within the first clarification spin, the amount of particles we get, when we first do that EV isolation, and then always compare the, say, AD brains alongside the neurological controls. Yes, you're going to see, so generally the proteomic composition, if you're just looking at general surface proteins, for example, generally they should be staying they should be similar. We do see differences in regards to number of EVs, and so that then does impact your downstream analysis. So then the question becomes, do you normalize to the number of EVs or to the protein? And that, I suppose, depends on, again, the question you are asking. I think at the end of the day is when you are doing these experiments is basically report everything that you do. We haven't found that digestion is adversely affecting the EVs we're isolating from neurodegenerative tissue. So that's a positive, but again, it's coming back to the good old-fashioned technique of basically measuring protein content, particle amount, but importantly, it's that digestion of proteins and doing the good old-fashioned Western blot.
Okay, any more questions from the audience? If not, we will close. I don't see any hand. Okay, perfect. That was a really great session. I would like to thank Professor Laura Vella again for the fantastic talk. And we will be uploading this recording in our YouTube channel. So please have a look at it, or you can share if you or your colleague missed out. And then with that, I would like to close our session for the day. Have a good day, everyone. And thank you to Poppy and Yi for moderating as well. Okay.
Thanks, everyone.